Hello and welcome to the Deep Sea Podcast, a punk take on a science podcast about everything deep sea. I'm Dr. Tom Lindley, curator of fishes at the National Museum of New Zealand, Te Papa Tongaroa, and with me as ever is the Professor Alan Jameson, Director of the Deep Sea Research Centre at the University of Western Australia and scientist for Inkfish. Any of our dumb opinions are our own and do not reflect the opinions of our respective organisations. Hello, mate. Merry New Christmas Year. Hello.
Evening, Tom. Happy New Year.
Morning here. But yeah, Happy New Year. You had a proper cold Christmas.
I did. Well, it's colder now, but yeah, I did it. I'm in the right hemisphere for Christmas, not the fake one. Not fake Christmas that you get down under.
My Christmas present this year was a barbecue, which I defiantly used in a tropical storm. So I've got a video I'll have to send you of just me being absolutely hammered by a rainstorm using my new barbecue because it's Christmas.
Yeah, well, it's snowing here now, so it's a bit more festive now. But Christmas is long gone. It's now the new year, 2026.
We had our Christmas party. We had our Deep Sea Podcast Christmas get-together.
We did. That feels like a very long time ago.
It does, actually. I mean, part of that is my first apology should be that this episode is going to be a week late, but it's that time of year. It's difficult.
It's Tom's special time of year, though, isn't it? It's not just that time of year. It's a special time of year.
I do love Christmas. But it was just lots and lots of other stuff that made this one quite busy. So the big show opened as well. The one I've been one of the curators of at Te Papa. So our big summer show, Breathe, Mauri Ora, opened. It was really nice. Did a nice bit of schmoozing. Nice themes of humans' connections with nature, and there's no hard boundary. We are all sort of a continuum of life. And it was really interesting talking to people from lots of different cultures who sort of, it spoke to them in different ways, and they had lots of different words for this level of connection that most cultures had spotted at some point. If you're in Wellington, come on down, give it a look. It's quite a chill and therapeutic little session. There's even a bit of meditation in there. It's a nice, just relaxing one. With Kate Blanchett. Kate Blanchett whispers in your ear in a forest and makes you all relaxed.
Really?
Yeah.
Is she actually in the forest, or is it just a recording?
It is just a recording. I'd love it if she just walked around the room and just whispered gently in everyone's ear. But we couldn't afford that. That's quite an undertaking.
I was going to say that's quite a big ticket item, having an earless celebrity 9 till 5 every day for 6 months.
Whispering in individual people's ears. Any other Christmassy news from you? How's the break? You're back in the UK, but back to Oz pretty soon.
Yes, fear not, Australia, I'm coming back.
Brace yourselves. Currently hottest place on the planet. So quite a change from the snow.
Well, I was walking back from town the other day and I figured I might actually be in one of the coldest places in the planet at the moment. So it's going to be a couple of weeks of extremes. Nothing planned, which is suspicious because I don't have any foreign travel really planned other than going back to Australia. Yeah, someone's going to call in the next couple of days and say Canary Islands, or
someone's going to send you somewhere.
I'm supposed to go to London tomorrow, but I suspect the absolute abomination of the British weather is going to kill that pretty quickly. So I'll be here for another week. In terms of song of the month, why don't we go for the national anthem of Liechtenstein?
Well, I did have a song of the month, but if you want the national anthem of Liechtenstein, that is an option.
I think we should, just to see what Spotify makes of it.
Oh no, don't just try and get me in trouble. Our listeners may hear the national anthem of Liechtenstein here, or they might hear It Is Deep, Bro by Matt Storer, who is an online comedian, particularly on Instagram, who did a nice little song about the deep sea.
Deep sea creatures, creepy weird freaks, huh? Heebie-jeebie species need up reaching every peak. Well, let's see your features under several kilometers. Your marine species, follow me for treatment. Tell this goblin shark that he's worthy of love. Good. Tell this gulper reel, you're glad that he's real. As you should.
Yeah, quite deep sea positive. It was similar to my blobfish rant. It was similar, like let's see you look good under all that pressure kind of thing. So it's quite cute. It's quite cute. So you might be hearing a little bit of that now, or the Liechtensteinians of you might be saluting furiously.
Well, I was thinking as we try and get around the whole Spotify thing, rather than having song of the month, we could just have national anthem of the month.
Public domain track of the month.
Yes.
Do you know what is public domain?
Songs. What?
Almost all hymns. We could make it hymn of the month.
Oh, we can't. We've just come out of Christmas. There's too much of that.
You've had enough.
You know, it's really funny with Christmas when they always go on about it's a magical time of the year and people talk, the magic of Christmas. Have you ever read what Christ said about magic?
Strong feelings.
He didn't like it. It's not a thing you should say, the magic of Christmas. He didn't like that. He's not going to enjoy that.
And the Puritans banned Christmas. It's funny that the original war on Christmas was actually led by the Christians.
Yeah. So we're not doing this for another 2026 years.
And certainly not starting it in late September. I want to say a big thank you to our patrons. As ever, we could not do the show without them. So thank you for those that continue to support the show. And to get into some news, there's been some stuff going on. A reminder as ever that we put a lot of the links and wider reading onto our website, so you can check out deepseapod.com to read a little bit more about any of these stories if they catch your eye. And quickly to follow on from the last episode, not quite news, but kind of news, we spoke to Sarah Mack on that one, the hero behind Skype a Scientist, and she's just released some really cute deep sea Valentine's cards, and all the proceeds will go to supporting Skype a Scientist, which puts real researchers in touch with schools and communities and sort of gives a very human face to science and helps us reconnect with communities. So we're not affiliated with Sarah, just that we share the same goals and love what she does. So there's a plug there, and we'll put the link into the show notes and on the website if you would like to get your Valentine's Day cards ordered nice and early. An unknown species of squid was spotted burying itself upside down and pretending to be a plant, but I'm going to put that bit in inverted commas because I'm not quite sure on that bit, but the rest of it's really interesting. So really unusual behavior noticed. So while studying the depths of the Claritin-Clipperton zone, an unknown species of squid was seen burying itself upside down on the sea floor. So although mud covering and burial has been seen in octopuses and cuttlefish and little bobtailed squid as well, and some shallow species, it's not been seen in deep sea squid before, which tend to be up in the water and pelagic. During the dive, the squid first appeared, then seemed to disappear, leaving the science team puzzled about where it had gone until they realized the squid was buried with its tentacles sticking out. So it looks like two fronds of seaweed sticking out of the seabed. The current theory is that squid was mimicking a glass sponge to attract crustaceans by burying its body with only its tentacles revealed. The thing that I have an issue with on that is we can't forget what the environment is like when we're not there shining lights on it. In complete darkness, maybe with bioluminescence going on, it's not going to look like that. You don't try and look like things. I think there might be something more complicated going on with sort of photophores here, or it is a way of escaping predation with the tentacles sticking out almost like cat's whiskers. So you hide yourself in the sediment, but if your cover is blown, you've got this early warning system of your tentacles hovering up. But that's just my opinion. So we're not quite sure why the squid's doing this.
Technically, I am mimicking a glass sponge in complete darkness. You just can't see me what I'm doing.
Well, by being into complete darkness, you are a glass sponge.
Yes. As soon as the lights goes off, you don't know what I'm doing. So, yeah.
There was a little bit of debate over what kind of squid this might be. The original article cited a whiplash squid, but there's a couple of counter hypotheses putting it in the Promoteuthis as a possible species. And we were discussing this on our Discord, and then Mike Ficcione of the Squid Phone fame chimed in with a little feedback on the article, which was the hypothesis that the unknown seafloor squid is mimicking another animal could be right, says Mike Bicchioni, a retired curator of cephalopods at the US National Museum of Natural History, but it could also be doing something completely different. We just don't know. I like Mike's down-to-earthness.
Yeah.
Dr. Bicchioni also isn't convinced that the quickly glimpsed squid is a kind of whiplash squid. He thinks it's more likely a promoteuthid, a rarely seen type of squid. And the authors actually said that identification is a plausible alternative, but without a physical specimen, we can't be definitive. So that usual tricky thing, an interesting piece of behavior in a very rarely seen squid.
A rarely seen 7-armed blob octopus filmed by underwater camera. I like it as filmed by underwater camera as opposed to
A sketch.
Anyway, there's a team at Mbari have encountered this 7-armed blob octopus at 2,300 feet, which is 701.04 meters, according to my distance converter. But this is the fourth time the organization have observed this species in nearly 4 decades, because of course Mbari work quite extensively off the west coast of America. So 4 times in 4 decades, that's once every 10 years you've seen this thing.
Considering how thoroughly they researched that area, this is probably one of the best pelagic deep sea study sites, and the fact that you can still see something at 10-year gaps. Like if you do that rarefaction curve, if you sort of figure out how quickly we're discovering things, like we've still got so much to go.
So apparently the female 7-armed octopuses can grow to considerable sizes, reaching 165 pounds. Not sure what that is in kilograms.
It's quite expensive.
Yeah, it says very expensive, yeah. Well, the males are far smaller, often no more than 8 inches long. So here we go. So despite its name, the species has 8 arms like other octopuses. In males, one arm, the reproductive arm, is kept tucked into a sac beneath the right eye, creating the appearance of only having 7. So it's not a 7-armed octopus at all.
It has a secret one in a pouch, in an eye bag.
Yeah. It's in some sort of imposter. So in this recent footage, the octopus was holding a crimson helmet jelly, which is consistent with past MBARI findings, and that these particular species prey on these gelatinous animals. There you go. 7-armed octopus has 8 arms. It's just a bit shy about its funny 8th one.
It's shy about its penis arm.
Yes. It's got 7 normal arms and 1 rude arm.
The rude arm. We
It's
should
got a rude
rename
arm.
it that. Yeah, petition to have it renamed that. Yeah, that's really interesting. I know vampire squid are detritivores when they get a little bit older, but it's interesting that something as high energy as an octopus can get by on jellies. Really not nutritious. It's like 1% protein. The rest is just water.
But maybe they just eat it because they like it.
Oh.
Maybe they've actually had their proper fill and they're just having like a bowl of Cheerios where they know it's actually of no real advantage to them, but they just fancy a bowl of Cheerios.
Oh, they've had
Maybe
their
that's
steak,
what
egg, and chips, but then they just have a little jelly
Yeah.
afterwards. Oh, okay.
Yeah, they've had their 5 a day and it's just a little late night snack. They could go without, but they do like a red helmet jelly.
I suppose it's pretty easy to eat if your esophagus passes through your brain.
Yeah.
For our dose of art, a former sub-pilot's art highlights the deep sea. Kerry Kirby was chief pilot for HURL, H-U-R-L, since 1981 and has completed over 900 submersible dives around the Hawaiian Islands and Central Pacific. And Kirby retired in 2019 and the HURL program has since ceased. So this was the big deep sub program out of the University of Hawaii, or affiliated with the University of Hawaii, wasn't it?
Wasn't it the Hawaiian Undersea Research Laboratory? Isn't that what HURL was?
Ah, that's it. I was struggling with the acronym. So in 2019, Kirby began creating precise illustrations of the historic wreck sites around Hawaii, dating from the time of the Hawaiian Kingdom right up to World War II. He created these images by referencing bottom maps and videos to recreate each site as they were found. Of course, when you're in the vehicle, you see things piecemeal. You see the little floods of your lights and the bit that's right in front of you, and then you have these seismic maps that put that at a greater scale, but I think he was working to sort of combine the two and almost mosaic it for a full view of the area. Kirby's also created work based on the Hancock Seamount and the Emperor Seamounts, which are quite famous research sites, which had been destroyed by trawl fishing and coral dredging. So his work gives real impact by going outside the sub and showing the whole area of impact, alternatively illustrating the massive coral communities that he had explored in 2016 to 27, which can be 2,000 years plus old, and using his work to document the destruction inflicted upon the areas. So we'll include some links. We like a little bit of deep sea art, especially for people who have been so moved directly by the deep sea. So you can have a look at that as well. And actually the deep sea art channel on the Discord is doing really well. I can't believe how talented some of the listeners are. There's some very cool pottery coming out. There's an I Am Your Tongue Now cup.
Oh.
Yeah, it's amazing. It's amazing. So it's a coffee mug that looks like a fish. And then as you drink it, a little parasitic isopod is in the bottom and you reveal it.
Oh, I like that.
It's very good.
That's good.
It's very good.
Yeah, cheeky. So the next story is zombie worms are missing and scientists are alarmed. Zombie worms being Ossidax, the bone-eating worm. So Ossidax are these things that they're technically polychaetes, right? And they live on whale bones. They've been in the deep sea for a long time. So the ones you can see tend to be females. The males are microscopic. They look like tiny little pink and white stripy plants that suck the lipid out of bones. And have been doing it since before whales. Apparently there's Ossidax scars on the old plesiosaurs. It's kind of cool. Anyway, this particular study found that whales that had put down in Barclay Canyon off British Columbia, Canada, had been down for 10 years and they brought them back and didn't find any. So it says this lack of Ossidax worm suggests that expanding oxygen minimum zones in the Northeast Pacific and beyond may be disrupting whale fault ecosystems. Interesting. Because we did one. We put down a whale skull for 440 days at 2,000 meters and it only had one. There was evidence that ones had been in and out. And I wonder maybe not the bone had been a bone too long. And if these things are there to live on lipid and there's no lipid left in it, then why would they go for it?
Yeah, when you've got to settle out of the water.
Yeah, because I've seen bones gone down before and they're absolutely covered in Ossidax. But that's a sort of natural event where either the bone has come from a fresh whale carcass or it's a natural one found on the bottom. But the one we had, we don't know when it died. So that whale could have
Trawled
died
up, wasn't
years
it?
and years. Yeah, it was trawled up and then we put it down, didn't get very much. It was maybe the one that was in it was the one that went in it and found it was empty and told all his mates not to bother.
Well, even though they're really associated with the sort of final phases of a whale carcass, the sort of terminal phase where all the flesh is gone,
Yeah.
maybe the signal of that flesh is important at the earlier stages. How are these larvae going to settle if there's not a good strong signal? That it could be the expanding oxygen minimum zones, which is definitely happening and acting as a boundary to certain species because they're not low oxygen tolerant enough to make it to the other side of that boundary. That would be a really interesting sort of result to that, seeing these changes.
Nice to see Ossidax back, though.
Yeah.
I'd like a good Ossidax story.
Very, very weird. There were some indications as well that it was affecting other, what would you describe them as? Not detritivores.
Wood boring bivalves. So apparently there was much lower numbers in the Berkeley Canyon than has been in other oxygen-rich waters. The new formations of oxygen minimum zones as a result of changing climates may well be shifting some of the deep sea communities.
I do wonder what the cues are. I do wonder how they find a new one.
I don't know, because the one we got out of our stinky minky skull, I gave it to Adrian at the National History Museum and he sequenced it, and it came back and it was Ossidax frankpressi, and the only other sample of that species came from Monterey, and our one was in the Rockall Trough off Scotland.
Well,
so there's two. I think there's one North Atlantic off Scotland and one off California.
Wow.
Go figure.
Okay. Whale tag is helping to decode communication in the deep ocean. So whales and dolphins are dependent on sound to communicate in the ocean. Tracking those sounds is difficult. They bounce around a lot in the ocean. It's hard to see which animal is making the sound and which one is responding, as it all just becomes a cacophony, especially when a whole pod is moving together. So a new open source biologger has been designed by the Harvard University team, seeks to solve this problem by recording sound and sensor data while being attached to the whale. Not only can the tags record and isolate sounds from individual animals, it actually has multiple hydrophones, so it allows it to triangulate and see where you can tell that the source is different. So it really is quite an advanced hydrophone. It can also measure pressure and calculate depth, acceleration, rotation, temperature, light, and even keeps accurate time, so researchers can line up these recordings to other data points at the same time, including using drones and fixed listening stations or even other tags. And what I really like is the team have released the hardware and software as open source, allowing other researchers to build on, adapt, and enhance it rather than starting from scratch. And I had a little dig into things they'd shared, and it's a really nice system. They've solved a lot of problems. It's quite repeatable. And to be honest, very different sensors could be plugged into this. So it's a really nice starting point, not just for cetacean biologists, but actually anyone looking for a deep sea tolerant logger. It's a really nice starting point.
Next thing's pretty cool. The next one's is people have uncovered some massive aggregation of deep sea eggs around an active volcano. This is off British Columbia, about 1,500 meters near what was thought to be a completely dormant volcano. But the ocean floor was covered with what is reckoned to be over 1 million oval-shaped eggs from what is believed to be the Pacific white skate. And so the geothermal activity came as a surprise to the scientists, which came from Fisheries and Oceans Canada in collaboration with Ocean Networks Canada, who were expecting cold, inert geology, and instead they discovered the first confirmed site of hydrothermal activity in Canadian water. So that's two big wins.
Yeah, that's huge by itself.
Yeah. And then the third win comes because apparently they also captured the first footage of a female skate laying eggs in the wild. Over 1 million.
Yeah.
That's pretty nuts. That must have been awesome, that. Very impressed.
Bathyrraja spinosissima, the big deep sea skate. The Bathyrraja tends to be the genus of the big chunky deep sea skates. But we've seen this in a few other species as well, with putting their eggs somewhere a little bit warmer to help them mature a little bit quicker. That makes a lot of sense that they're brooding their eggs here, and considering the low density of deep sea animals, this is a huge amount in a small area. Highlights the risks of sort of harming a nursery ground and how that might have impacts on the species because they will congregate in one place. And so this is obviously quite an important spot for them. Should we get into an interview?
Yes.
I struggle to segue from any of the news stories, but I think that is part of the significance of this interview, is that it's something incredibly important, really, really cool, but very rarely spoken about outside of direct benthic deep sea circles. So what if we were to tell you that there are giant single cells on the seafloor, with a single cell being up to over 20 centimeters long, and it builds that shell from a variety of materials and is a crucial part of the deep sea community, yet we hardly talk about them at all. I think the general public and people listening to this, I'd be really curious to know if you've ever heard of these before, but these are the giant Testate Protists, the Foraminifera, xenophyophores, and gromids. And when it comes to talking about these, only one person springs to mind, Professor Andrew Gooday of the National Oceanographic Centre in the UK, who spent decades studying these groups and is the prominent researcher on them. I worked with Andy on Ecomar, and I think you still send the odd picture his way to help ID these strange critters.
Yeah, we talk to Andy quite regularly because there's an unbelievable difficult group of organisms that just really struggle with them. Because certainly down in the deep sea, the very deep sea, the Hadal stuff, there are so many. There's some states we go to and it's just thousands and thousands. I think the last we counted was something like 8,000 on one dive. And then you go to anyone and say, what are these? And they go, I don't know. Are they forams? Are they xenos? Are they communications? Are they gromids? Well, no one really knows. And you've got to collect them and be able to tell. But even then, it's quite difficult. And they're just such a difficult group of animals that I don't know. So I do speak to Andy from time to time because I can't make head or tail of it.
Even with a sample, some of them you still can't tell. It can be hard to find tissue.
But you're right, I'm surprised more people are not genuinely into this because it is so weird.
They're very cool.
They're so difficult, but they're so ubiquitous as well. I mean, they're everywhere and they come in every possible conceivable shape and size. They don't seem to care at all about pressure. I think they're actually one of those ones where it goes beyond curiosity into this is just really difficult now. It's like the uncanny valley, but it's like how effort versus reward. And I'm glad that Andy and others have taken on this group because I don't think I have it in me. I don't have the fuse for this group.
They're very cool to
me up the
learn
wall.
about, but they're hard to study, certainly.
I find them fascinating. I genuinely do. And I wished if I wasn't so busy doing other grown-up things, I wished I could spend a bit of time really
getting your head around
these things. But yeah, we need to find some new young buck who's willing to spend the next 45 years getting really into this. And we need to start collecting them more. But yeah, they're a really big one. It takes a special person with a special disposition to get into the xenophile force. And thankfully, Andy Gooday is just that guy.
So coming to you from the 3rd International Conference of Biology, Geology and Environment of Hadal Trenches, accosted Emeritus Professor of the National Oceanography Centre in the UK, which is one of the big hubs for UK research, particularly into the deep sea and Antarctica. Andrew Gooday, our audience is big fans of the deep sea, but I bet a lot of them will not even be aware of the group that you work in, which is the Foraminifera. How would you describe them? How would you sort of begin for someone who's seeing them for the first time?
The Foraminifera, or the forams for short, which is a lot shorter and easier to say, are protozoans or protists, as they're called now. So they're single-celled organisms, and most species build a shell or technically a test, which is a protective covering. And the shell is made out of sometimes, in some cases, calcareous material, so calcite, usually calcite. Sometimes it's composed of particles which they pick up off the seafloor. So that type is called agglutinated. So those are the agglutinated foraminifera. And then some have an organic test wall, which is transparent. They live in aquatic environments. So you find them at all depths in the oceans, from intertidal rock pools, shallow water, right down to the deepest part of the ocean. And some types also occur in fresh water. They've even been found in soil, which is quite amazing, although they're probably living in sort of damp conditions, damp soil and damp leaf litter and in damp moss and things like that. Most people associate the foraminifera with the oceans. I work mainly on deep sea forams. So they have a cell body. The most remarkable thing about them is their pseudopodial system. So they have these like threads of cell material, and they use that for all sorts of functions like, obtaining food, building the shell, respiration, production, and these pseudopodia, as they're called. Pseudopodia just means false legs. Pseudopodia are remarkable. They have unique features, a very sophisticated system of proteins which enable them to move very quickly and reform and make different patterns on surfaces.
One of the most striking elements is how large they can be. This is a single cell which is of a scale we usually associate with an animal.
Forams span an enormous size range, or almost larger than any other sort of group of organisms. So the smallest ones are just a few tens of microns. You need a very powerful microscope to see them. Those are the smallest ones. The biggest ones, which are the ones I'm particularly interested in, that we're going to talk about particularly today, are called the xenophyophores, and they grow usually up to 10 centimeters, 15 centimeters, but the very largest ones can exceed 20 centimeters. And this isn't the actual cell. This is the shell that the cell builds. So the cell actually, the volume of the cell is much smaller than the shell in which it lives. The structure that it builds can be, up to, more than 20 centimeters. And so they're in the Guinness Book of Records as the largest protozoans, the largest single-celled organisms. They live in the deep sea, and the shallowest records are from about half a kilometre, about 550 metres off the coast of New Zealand.
Yeah, we do seem to have a lot of stuff that looks deep sea but shallow. I see things snorkeling that I associate with much deeper water, like we've got elephant fish, chimeroids like you can snorkel with.
Well, I think these really shallow records are from the Chatham Rise.
Oh, yes, yeah.
Isn't it a sort of a continent or a sunken continent?
The whole sort of area is a continent the size of North America, but it's just underwater.
Yeah, so those are the shallowest ones. Then you find them in all sorts of places. And they're not common everywhere, but there are many places where they are common. A lot of them on seamounts. So if you have a slope, you tend to find them. Probably because on seamounts or sloped surfaces you get currents, and some of them feed on suspended particles. They like that sort of place. We found many specimens in submarine canyons. Off the coast of Portugal, there's a canyon called the Nazaré Canyon, which comes up right close to the beach, and it ends up on the abyssal plain, 4.5 kilometers deep. It's called the European Grand Canyon. And at the bottom of the Nazaré Canyon, we found lots and lots of xenophaphores. They were up to about 10 centimeters, some a little bit more than 10 centimeters. And they presumably were there because currents were coming down the canyon.
They're like the food to be brought to them, really. They're like areas of current.
Yeah. And then you find a lot of them on abyssal plains. You don't find so many currents on the abyssal plains. So it's kind of strange that in some places they're attracted to currents, and other places they live in not a very strong current.
Are they particularly strong? They've always looked very fragile to me. I've never wanted to touch them because they look so fragile. Is there sort of an optimum where they want it to bring food to them, but they don't want to be blasted apart?
Yeah, they vary. Some are quite robust, surprisingly robust. Yeah, but most of them are different degrees of fragileness. Some are extremely fragile. So it's very good to, if you can collect them using an ROV or a manned submersible, if you can take photographs of them on the seafloor in their life position, perfect condition, intact. And then when you collect them, there's always the danger that you'll damage them while collecting them. And then when you get them back on the ship, you have to be very careful, usually. But usually they do survive okay.
How do we preserve them? Do we dry them? Do you preserve them in alcohol?
You can do all three of those things. You can preserve them in alcohol. We usually preserve them in formalin. You can also dry them. Yeah, many of them are robust enough to dry, and in some ways that's probably the best way to preserve them in the long term. At least most of them are agglutinated. They make a shell test out of particles they pick up off the seafloor. It depends what material they use. If they use sort of sand grains or shells of planktonic organisms like radiolarians and planktonic forearms they use, then they're quite rigid. Some of them are made of mud, very fine grained sediment, and those I don't think you would want to dry them. Those are particularly delicate. So where else do we find them? We find them down to the deepest places in the ocean. So they've been photographed in the Challenger Deep at 10,900 meters, which is the deepest point in the ocean. And we know that they're very common in trenches, in submarine trenches, these very deep trenches, which include places like the Challenger Deep. There's not much of that is published, but people have observed them in trenches.
In the areas where they are abundant, they are really, really abundant.
Yeah, exactly. Yes. That's right. They're often the most abundant organisms that you see on the seafloor. People are very puzzled by them. Now, often people don't recognize them for what they are. I think they're becoming a little bit more famous now, but still people see them and they don't know what they are.
Some of the morphology that you showed today, the variety in it, me and a few other people turned to each other saying, like, I would misidentify that. I'm sure that was a bryozoan, I'm sure that was an encrusting sponge. The ones I've seen most often, which I describe as looking a bit like an onion bargee, they're sort of spherical, and I think you call them reticulated.
Yeah, there's a reticulated.
There's not much else that looks like that, so I'm sort of confident with those. But some of the forms you showed today, I was like, oof, I would have mislabeled that for sure.
There's an enormous variety of morphologies. Some of them are spherical, some of them are flat plate-like things, some of them are contorted plates, some of them are tubular, some of them make reticulated tubes, some of them, as you said, are reticulated, and they can live on the surface of the sediment. They can also attach to hard substrates. So in the Pacific, in the equatorial Pacific, where we have a lot of polymetallic nodules, they sit on the nodules. So I think some species are specialized to living on hard substrates, others are more specialized to living on soft sediments. Some of the ones that live on hard substrates, they can stand up. They're often fan-shaped. They look a bit like a fan on a stalk with a sort of semicircular upper part, which is plate-like. And we think that they pick particles from the water. They're suspension feeders.
So they're following the model that we see. If it's open sediment, you've got deposit feeders, and then as soon as there's a hard substrate, you can lift some sort of feeding apparatus into the water. You take advantage of that, and no wonder they end up looking like the same things that do that. That's the optimum shape to be. So they start to look like a sponge.
Yeah. Some of the morphologies are very similar to bryozoans, for example, and to sponges. And I think this semicircular fan at the top, it's an efficient shape for deploying pseudopodia. So the pseudopodia, I think, will be deployed around the margins of this fan, just stick up into the water, and they can intercept passing particles. The ones with a stalk, they have sort of like root-like structures, which extend out across the surface, because they must get the agglutinated grains, the grains they make to use the test, they must get them from the sediment. They presumably access the food in the water column from the fan, and then they have these roots, and they have pseudopods which are deployed from the roots and spread out into the sediment
to scavenge the raw material.
Yeah, the raw material to make their house, to make their test.
It's like a quarry. They've got a quarry that's mining.
That's right. They have extraordinary adaptations. Shall I tell you a bit about the internal structure? If you break one open, or you collect it and you carefully chip away the test, if you're lucky, in some species, it doesn't apply to all species, you will see the cytoplasm. The cytoplasm is the cell material, and it forms a branching structure which ramifies throughout the interior of the test. So that's one strange feature. Another strange feature is that they accumulate large masses of waste material. So part of the interior of the test is occupied by this branching threads of the cytoplasm. The rest of the interior of the test is occupied by accumulations of this waste material. And the waste material consists of little pellets. They're like fecal pellets. And they're just masses of these fecal pellets which fill up much of the interior. You can also get agglutinated particles on the inside. Sometimes they have an internal architecture as well. And in that case, the threads of cytoplasm weave between these internal pillars, and then these masses of waste material also arrange themselves around the internal architecture of the test. And we don't know why they do it. It could be that they're filling space, that they're bulking out the test to make themselves big.
And is that about a feeding surface area? Is that about becoming more efficient?
No, I think it's about being big. They seem to have this large volume, small biomass, or relatively small biomass type of organization. And that seems to be associated with oligotrophic conditions, food depleted conditions where there's not much food. It's a kind of organization which is found in other forearms in the deep sea. I mean, that's one idea. There's another idea, which is that they use these waste pellets to farm bacteria. The bacteria grow up on the waste pellets, and then they feed on these bacteria. I mean, it's a very nice idea, although there's no evidence at all for it. So it's probably wrong. I mean, I think they use it to bulk out the test, personally.
What are the pellets actually made of?
They're made mainly of clay material, but they also have organic stuff in them. You see curious sort of folded membranes and the remains of organisms like radiolarians or bits of planktonic foraminifera. It's obviously sort of ingested particles, but most of it is clay.
Right, so really fine material.
Yeah, fine-grained material, which they either get from suspension. I mean, you might get clay particles in suspension, or they get it from sediment.
Is there any sign that they're selective in what they pick up to make their test?
Yes. In different places, they choose different things. A lot of the ones in the abyssal Pacific that we've worked on a lot are made of volcanic ash or jagged, irregular shards of silica sometimes with bubbles in them, which looks like bits of volcanic ash. So I've worked recently with the Japanese on xenophyophores around the Japanese islands, and they use this what looks like to me like volcanic ash. I would need somebody to confirm that, but anyway, they're siliceous particles which are very jagged and irregular. There's one particular group, which is characterized by having organic threads in the test. So the test has agglutinated particles, but it's quite soft. The test is held together by these organic fibers. Usually the test is rigid. It's like a biscuit.
It would crumble if you broke it.
Yes, it crumbles, exactly, yeah. But this particular group, they have a soft test, and it's like a rag. It's like a wet rag. You can fold them over, and they don't drape, and that's because they have these organic fibers which ramify the whole of the test and hold the whole thing together so that it's flexible. And these organic fibers are made of a protein which is very similar to silk. So they make silk.
So that's a really complicated molecule. They've not just secreted a mucus and stuck everything together. That's complicated and strong.
Yeah. Well, the fourth strange thing about them internally is that if you take out a strand of the cytoplasm of the cell material, put it into a scanning electron microscope and look at it, you will see lots of crystals. The cytoplasm is often full of crystals, mineral grains, and they're usually barite, which is barium sulfate. For some years, there was a debate about the origin of this barite. Some people thought that it was made by the xenophyophore, and the idea was that for some reason they took on board barium, and barium in a soluble form is toxic.
It's a form of detoxification.
Now I'm pretty sure they get it from the sediment.
Which might mean they want it.
They get it on purpose for some reason. So you find barite in the sediment, crystals which are almost identical to the crystals that you find in xenophyophores. And there's been quite a lot of work on this sedimentary barite because it forms in the water column inside organic aggregates. These sinking organic aggregates. There's a microenvironment inside, and for some reason, the chemistry I don't understand, barite precipitates inside these organic aggregates.
Is that the marine snow?
Yeah, marine snow.
Right.
Yeah.
Wow, okay.
They reach the floor, the marine snow decomposes, and so they accumulate in the sediment. Sedimentary barite is used as a proxy for productivity.
Wow, because it's almost the skeleton of a piece of marine snow. It's the bones.
Yeah, exactly, yeah. And the thing about barite is that it's heavy. It's a dense mineral. I think they pick it up because it's a heavy mineral. And the reason I say that is because there are some other forams which belong to the big group called the monophthalamids. So the monophthalamids are primitive forams, which are usually single-chambered. That's the defining characteristic of them. It's a very big group, very, very extremely diverse. And so the xenophyophores belong to this group of monophthalamids. And there's some other monophthalamids, much smaller, which pick up other heavy minerals, like magnetite, which makes them magnetic. Zircon, they will pick up. Ilmenite, which is a titanium mineral. But these minerals come from land. They're washed into the sea from land because they come from various sort of metamorphic and igneous rocks. So the forams that accumulate these things inside the cell, they're coastal species or they live close to land. But in the open ocean, the only heavy mineral that you find is barite. So that's one reason that strengthens my belief that they get the barite from the sediment and accumulate it because it's a heavy mineral. They've got this love of heavy minerals, which they share with some coastal species. And why on earth do they do that? I have no idea. Well, I've got some vague ideas. I mean, some of the small ones, which do this in shallow water, they have a simple test which has an aperture at one end. It's oval with an aperture at one end.
They look like irons almost.
Yes, yeah. There's a genera called Samophaga, which means sand eater. And so Samophaga is the classic one that accumulates these grains. So they have an aperture at one end, and they tend to accumulate these heavy minerals at the aperture end. And so it could be that they weigh down the aperture so that it's embedded in the sediment.
It's like the kid's toy, the Weeble Wobble.
Yeah.
The little people that are weighted on one end, so they're always the right way up.
Yeah, exactly. Yeah. But that's not the case with xenophyll. I mean, they're far too big anyway. I think it may be to make the cytoplasm rigid, because the cytoplasm forms these branching strands. If they didn't have the barite crystals in them, it might flop around because they pack it out. I mean, there's huge numbers of them. In the SEM, it just looks like a strand of barite. You can't see any cytoplasm. It just looks like this, thread of barite crystals. I think it is possible that it makes these threads of cytoplasm more rigid so that they will maintain this architecture.
So they're building a skeleton. They're building bones.
Yeah. It's a kind of skeleton. Yeah. They're quite remarkable, and they have all these strange features. But on top of that, they're extremely important. They're one of the dominant large organisms.
It's not an oddity. It's not some fringe thing. It's very, very alien. It's very unusual in terms of what we think of as life, but it's a linchpin of the deep sea.
Yeah, and they're important habitats for other organisms. Because if you break one open, you find all sorts of creatures living inside. There's worms and there's crustaceans and ophiuroids, brittle stars, which are curled up around the base. There's a real affinity between ophiuroids and xenophyrophores, and I think it's because organic matter collects around the base of the xenophyrophore. There's a kind of a moat, and organic matter collects in there.
They're big enough to alter the boundary layer. So they're going to create a little microhabitat.
Yeah, that's right. And so they're like little reefs. There's a lot of them on the coral mounds of Scotland, the Darwin mounds.
Oh, right, the Coldworth coral mounds.
Yeah, so they're very abundant on those. And in fact, they've been designated as vulnerable marine ecosystems, VMEs.
In their own right.
Yeah, in their own right. They don't occur with the corals, but they are a VME.
Good to see him get some recognition.
Yeah. Bit of kudos.
Do we know what the lifespan is?
We were lucky enough to photograph 3 specimens on the Madeira Abyssal Plain off northwest Africa a long time ago, in the 1990s. And this was a time-lapse camera system called Bathysnap, which goes down to the seafloor and it stays there for up to a year. And it has a camera which takes a time lapse. And just by chance, it happened to be pointing at 3 xenophyrophores. And they started off quite small, and they increased their volume by 10 times over the course of, I forget quite how long it was. It was almost a year.
Oh, wow, that's quite quick.
So they grew up very quickly, and you could see something about the growth process. The remarkable thing was that they grew in a series of steps. So they were quiescent for a period of about 2 or 3 months, and then they suddenly grew, and then they were quiescent, and then they grew again. And there were 3 of these growth phases.
Within a single year?
Within a single year.
Right, so it's not tied to the bloom, but it feels like they're hoarding resources and then building and then hoarding resources.
Yeah. I mean, that was published in 1993, and the reviewer said there must be a cue. There must be something which is telling them when...
All 3 were in sync.
All 3 were in sync. That was the strange thing. You could argue that they were combining feeding with test growth. So they would collect a lot of sediment, they would use the organic matter to feed on, and they would incorporate the agglutinated grains into the test. But although why they should do it in a sudden burst, I'm not sure. It doesn't quite make sense.
It reminds me of something that malacologist Kerry Walton mentioned, which was about snails growing in sort of jumps, adding a whirl in jumps because the intermediate phase was more vulnerable to predation. So this kind of growth makes me feel that the interim is somehow not optimal. And so you want to get through it as quickly as possible. It would be continuous growth if it didn't matter. So the fact that they seem to hoard resources and then make a very decisive step makes me think that you don't want to be in between.
We have no idea what they're really doing. They're clearly picking up sediment because you can see them doing it. In one of the phases, there was a ramp of sediment off to one side, and it was there for a very brief time, and then it disappeared. Collecting it in, scooping it up, and then very quickly incorporating it. And during the incorporation phase, the there was an ornamentation, there was a structure on the surface. They were reticulated, and the reticulations disappeared. So they're obviously moving particles around on the surface. And then the reticulations reappeared, and there was an extra row of reticulations at the base. So they were adding the new stuff at the base, jacking themselves up. This is probably how at least some other species grow, because you often, on these fan-shaped morphotypes, you see growth lines, concentric zones. So you have a semicircular fan, and there's semicircular growth zones within the fan. So I think they grow in a series of jumps.
Based on how large you knew that species got and the sort of growth rate, which is really lucky from a time-lapse camera, that there were 3 musketeers in the view, what sort of lifespan do you think they would have?
I think a few years, maybe. Yeah, I think no more than that, yeah. The really remarkable thing was that all 3 grew together at the same time. It wasn't exactly the same, a few days difference, but they were more or less synchronized. Is there some connection? Is it like in a forest that there's some connections?
The Wood Wide Web kind of thing,
yeah. Connections between the trees. Is there some...
The chemical cues they do,
they're giving off, not pheromones, but some sort of chemical signal, yeah.
How do they reproduce?
I mean, it's not really known, but classically, 4-Ams will have an alternating life cycle where they have a sexual generation. They produce gametes, which form, to form a zygote, which will build a chamber and then grow up into a large adult. So they have a sexual generation which alternates with an asexual generation. So that the adult, which develops from a bisexual reproduction, the cytoplasm fragments into little tiny offspring, which leave the test of the parent and go off and live independently. And there's some old literature claiming to see gametes.
What do the gametes of a single cell look like?
Yeah, I think they're flagellated. I'm not sure.
But we just don't know is a totally fine answer. Thinking of this little trio in there, and is that synchrony, you don't want to waste resources releasing gametes when you don't have a sexual partner. So are they signaling to each other? Is there some sort of Zeitgeber, some sort of time cue that is letting them know?
Yeah. The other interesting thing about growth is that in 1989 or so, there was a big volcanic eruption in Indonesia, Mount Pinatubo, quite a famous eruption, and it produced huge volumes of ash. And the ash blew out over the South China Sea and were deposited down to abyssal depths. There was a project to study the recolonization by some 4-Am people to study the recolonization of this ash layer. Among the pioneers were xenophophores. The xenophophores grew up very quickly on this ash layer. So they seem to be pioneer recolonizers, which is consistent with their rapid growth. So they must be able to spread far and wide.
They couldn't just be budding to recolonize a whole area.
Yeah, so they must have a dispersal phase, which is presumably zygotes. There must be loads of these zygotes floating around, and some happen to land on this ash layer and grew up, and they were the dominant thing on the surface of the sediment.
Are they mobile at all?
Almost certainly not. You never see any track, associated with them, and they would be very unwieldy.
Yes, because their biomass is such a small proportion of the thing they've made.
They're not built for movement, but maybe when they're tiny, I don't know. Yeah, maybe if they find a good spot when they settle, maybe they can adjust themselves a little bit, and then they get too big to move. I don't know. That's just pure speculation.
We only catch glimpses of them, and there's so much still a mystery.
They're fascinating things, and they're difficult to work with. But we've got a lot of sequences. Oh, I say we, my genetic colleagues have.
Humanity.
Yes, yeah.
As a collective.
The other thing about the cytoplasm is that it's multinucleate, so there's lots of nuclei. So they're a little bit like slime molds. They've been compared to slime mold.
But it makes total sense that you couldn't run such an enormous disparate cell on a single nucleus.
Yeah, although some big 4-Ams only have one nucleus.
Really?
So,
Oh, I didn't know that. I thought they were all multinuclear.
Yeah, no, there's some big, tubes, which 10-centimeter tubes, which have only 1 or 2 nuclei.
So their internal transport network must be incredibly complicated as well. They're very charismatic. We don't give them their due. They're really important. They're really interesting, and we don't talk about them enough. And I'd be curious to hear how many of the listeners who are super into deep sea will be hearing this for the first time. They're such a fun group.
Yeah, no, I think they should get some publicity and some fame and benefit for their efforts at being amazing.
How on earth do you go about sort of separating them out? I'm guessing test structure is the big thing.
Yeah, the taxonomy is pretty chaotic. The classical taxonomy is based on the test structure. So there's a kind of hierarchy from tests which are kind of solid. And then the next step, there's different degrees of test architecture, so increasing complexity. And the simple kinds are solid, homogeneous structures. The most advanced forms have hollow interiors. The taxonomy, the classical taxonomy, is based on the place along the spectrum of complexity in test structure.
We believe they've developed towards increased complexity.
Yeah. But xenophyrophores are, well, relatively easy to sequence, probably because they have a lot of nuclei. And the genetics just does not agree with the morphology.
Yeah, that happens a lot.
Yeah, there's some correspondence, but there's a big debate with my genetic colleagues at the moment about quite what to do about revising the taxonomy of xenophyrophores. I think we probably need even more sequences. We have a lot. We have more sequences for xenophyrophores than we do for any other monothelenids, any other of this group of primitive forum. You certainly get groupings. The fan-shaped ones which live on nodules come together in a nice compact group. So there are very similar morphotypes which are genetically distinct. So the genetics is very useful for backing up these very subtle differences in morphology. But one of the big problems is that there are some classical genera which were described in some cases in the 1800s, some more which were described in the 1970s, before genetics. One problem is that some species which have been discovered more recently have been fitted into these classical genera. Often we have sequences for these more recently described species, but what we don't have are sequences for the type species of those genera. And as a result, species which have been put in genera like Samina are scattered across the xenophyrophore tree. But we don't know what Samina is genetically because it hasn't been found since. And it's the same with Syringamina, the Syringamina that we saw at the Ecomar. We have sequences for things which look a little bit like Syringamina. I doubt that they are the true Syringamina. The type species was described by Brady in 1881, and it actually occurs off the Scottish coast. It's this one which is a VME. Yeah, that's Syringamina fragilissima. And the one that we found at Ecomar is very similar. I think it's probably a different species. And Syringamina fragilissima from off Scotland has been collected, but nobody's ever preserved material for genetics.
These original descriptions, do we have types or do they tend to be illustrations?
Most of them were described by Haeckel based on Challenger material. And most of the species described by Haeckel came from the equatorial Pacific, but we haven't found them again, even though xenophyrophores in the equatorial Pacific are well studied. But I haven't seen anything that looks anything like Haeckel's illustrations. And that may be because Haeckel described them as sponges. And so there was a debate for some time about whether they were forams or sponges. And he happened to be working on the most sponge-like of them, whereas Brady described the first xenophyrophore and he described it as a foram. And he was right. But Haeckel described all these Challenger xenophyrophores as sponges.
But the morphology holds and that is still the type description, but the Hemut kingdom?
Yeah. There's no doubt that there's xenophyrophores. I mean, he made up a lot. He obviously saw the cytoplasm and he interpreted the cytoplasm as being a symbiotic hydroid.
Oh, okay.
And he has lovely illustrations showing these wonderful symbiotic hydroids, which are intergrown with the sponge, in quotes. And it was all, his imagination after these early species were described. And they were established as a distinct group. So Haeckel described some, Brady described one. And then there was a guy called Schultze, I should pronounce it. He got it right. He recognized them as protozoans, at least. he published a monograph based on the collection that he had. And then things went very, very quiet until 1972 when Ole Tyndall published this monograph. And since then, they've been getting more and more familiar. But as you say, they're still not as widely known as they should be.
They're also very useful as a sort of dating species in sediment. Is that, or is that the smaller ones? Sorry.
That's the smaller ones. I mean, most forams are studied by geologists, and they study them because they're excellent proxies for reconstructing conditions on the bottom of ancient oceans. So they can tell you about oxygen, and the calcareous ones have isotopes in their calcareous tests.
It's a little time capsules.
Yeah. So some are planktonic, so you can study the oxygen isotopes, and that tells you about the temperature. And the ones on the bottom, you can, the carbon isotopes tell you about, food inputs and so on. So they're used as proxies, both the formal information and the elemental composition and the isotopic composition of the shell, the calcareous shells. But you can only do that on calcareous shells. You can only do the chemistry because they have a really an outstanding fossil record. They're the most abundant organisms in the deep sea paleoceanographic record. There's nothing else really. There's nothing else benthic. There's plenty of planktonic things like radiolarians and planktonic forams and diatoms and coccoliths, but benthic forams are the only benthic organisms which are preserved in large numbers in the paleoceanographic record. So they're very important. And for that reason, it's mainly geologists that study modern faunas because they want to understand the ecology of modern faunas in order to interpret the fossil record. But because of this, they concentrate on the fossilizable species. So the xenophyrophores and most of the other monothalamids are very delicate. The monothalamids, which are very abundant on the abyssal plains, for example, they're very delicate. They won't be
preserved. Yeah.
So xenophyrophores have no proven fossil record. There's been various claims for fossil xenophyrophores. There's a trace fossil called Paleodictyon, very famous trace fossil, which has been found in modern sediments, and it goes right back to the Cambrian. It's a very hexagonal system of a sort of network of hexagonal patterns.
I think I've seen this
on the seafloor, and it's been suggested that they were made by xenophyrophores, but they occur on abyssal plains in several different oceans. We've been found in the Atlantic and Pacific and Indian Ocean, but there are no xenophyrophore remains associated with them. There was a big study of them in the 1990s, I guess. So there's a guy called Peter Rona who made a lot of observations of them from the Elvin submersible, and he cored them. He collected them in push cores very carefully. He sent the push cores to various specialists around the world, to sponge specialists, because one idea is that there may be sponges making these hexagonal patterns, and he sent them to other people for genetics and for microbiology and so on. And I got a core to look for xenophyrophore remains, and there was nothing. And I don't think they're xenophyrophores. At one time that was a popular idea because there are some tubular xenophyrophores which make a kind of hexagonal pattern. They're infaunal. They live within the sediment. Yeah, they form a hexagonal pattern. We've seen some similar ones in the Clarion-Clipperton zone in the Pacific. But the Paleodictyon pattern is extremely regular. It's almost sort of geometrical. At one time, when these patterns were first seen in the Atlantic, people thought they might be some sort of something artificial, something put down on the seafloor by
a lander feed or something.
A sensor, maybe a hydrophone of some sort, or some kind of artificial structure which was put down by the military.
Somebody knows exactly what that is, but they're not telling.
Yeah. But because they're so regular, although xenophyrophores, some of these infaunal xenophyrophores do make these hexagonal patterns, they're not really absolutely regular. And also, I think then Paleodictyon would be a fossil. There's a question of whether it's a body fossil, whether it's a structure, or whether it's a trace.
Right, which is just a shape left in the sediment.
Yeah, so a worm or a crustacean or something like that making it, building these tunnels in this hexagonal pattern as a very efficient way of feeding. And I think that's most likely. But nobody has ever found whatever's making it. It remains a mystery.
Oh, that's a great one.
Yeah.
I'd have to do some digging into that, especially if it's a deep sea habitat.
Yeah, there's quite a few papers on it now. There's been a bit of a flurry. Yeah, we had a paper in Scientific Reports a couple of years ago on, because we found some on the Aleutian. There was a German cruise in the Aleutian where we found these patterns, these paleodictyon patterns on the seafloor. They were originally thought just to be in the Atlantic, but now they've been found all oceans. Yeah, we saw them in the Antarctic, actually, in the Weddell Sea, but that was never published. I mean, there's a lot of other things to talk about, but about forams and monophyllamids and, well, gromids, of course, but that would be another podcast.
Part 2.
Yeah, gromids, that's another story altogether. But the key points, I suppose, are their size, enormous morphological variety,
Their importance.
their just sheer abundance and importance. Yeah, the fact they're confined to the deep sea, that they don't occur in shallow water. Why don't they occur in shallow water? Yeah, I don't know. There are very large forams in shallow water. They're calcareous forams that grow up to 15 centimeters. Yeah, so
But the xenos don't.
But the xenos don't occur in shallow water. I don't know why. Oh, there are some naked xenophoraphores.
Really?
Yeah.
Just a giant cell.
Well, the giant cell and the masses of waste material. The body is made out of waste material. That's quite a recent discovery. There's one in the Java Trench, and I know Lisa Levin found one. Yeah, a lot of this is unpublished. There's none which are described, but Lisa found one on a seamount and illustrated that. But yeah, that's really strange, having a body made out of feces.
It's pretty efficient.
Yeah.
Turn a problem into a solution. And to be honest, sediment is often made up of waste, so why not use your own rather than scavenge it?
Yeah, exactly, yeah.
Well, thank you for having a chat. This is really nice.
It's a real pleasure.
Have you ever come across paleodictyon, either in your research or looking at the bottom?
I saw your notes earlier and I thought I recognised that word, so I googled it, and then I saw a preview of a picture and I thought I've just seen that in Antarctica in the deep sea.
Really?
But then, but it's not.
Oh, okay.
I've got something that looks a lot like it, but in the Antarctic photos, there's 3 different versions of this thing we've seen, and it's the whole ice raft debris, right? There's these black rocks that sit around and they've fallen from icebergs and glaciers. So the whole seafloor is littered with these things. Each one has one of these on it, and it's a white stripe. And that stripe is either lots of little eggs, well, not eggs, but lots of little gelatinous spheres, or it's like lots of little hexagons, or lots of little cups, all gelatinous, and they're like smeared on the side. It's not an animal that's moving. So I thought there were eggs, and then someone said it could be something else. And I sat one night a couple of months ago and I was really bored and just took all the photographs and said, those are all the ones that look like that. Those are the ones that look like that. Those are the ones like that. And they did have that kind of structure about it. When I saw that, I thought, oh, Jesus, maybe it's those. But actually it's not. It's a whole different, I think they're talking 2 different things. It's just a symmetry, this sort of repeating pattern is quite intriguing. But I'm not quite sure what they are, and I'm not sure what we're seeing in Antarctica either. I figured it because there's so many squid and octopus kicking around, I figured it's got to be something to do with them. They've laid their eggs on something, and maybe once one animal puts its eggs on this rock, everyone else leaves it alone, and that's why there's only one on each.
Yeah, they quite often coat them in something a bit nasty.
Yeah. But I know it's not that, but that's when I saw the little thumbnail come up at Google, I was like, maybe that's what they are.
Brain tingles.
But it's not.
I just can't believe how uniform it is. It looks like a trainer print. It looks like somebody's stepped in the mud with their Nikes, and it's just this perfect honeycomb sort of pattern. Wild that they have samples and we're still not sure. It's been scooped up and we can't find anything. Because of course everything in the sediment's alive anyway, so it's not like you don't get any signal. It's you get loads of stuff and, well, which one made it? I thought really cool. That's a really good mystery. We're not even sure if it's a trace fossil or a body fossil. We're not sure if it's a burrow or actually the animal. This stuff's great.
Ancient civilization art.
Oh, ancient Nikes.
Yeah, that on Discovery Channel could have been made by ancient aliens in the deep sea.
Nazi ones. Nazi
Of course they're Nazis. Of course they're Nazis. Who isn't
That's
these
a
days?
given. That's who isn't these days.
Hey friends, Megan here, back with one more update on the Unseen Ocean events we're hosting next month in Juneau, Alaska. Can you believe it's going to be February next month? I know I can't. I'm definitely spending a lot of time in the studio these days trying to get as many paintings done as possible. It's not hard to find inspiration for paintings in the footage taken by NOAA off the coast of Alaska in 2023. In fact, it's pretty hard to narrow down what I actually have time to finish before the show. since each square foot of the ocean floor seems to be utterly teeming with life. Sponges, corals, shrimp, brittle stars, amphipods, polychaetes, and uncountable others are crammed into every available space. Many times I find myself lost in the reference images, locating a new organism every time I take another look. It feels like participating when you get to look at an image and find hidden organisms. It creates a sense of connection and fascination, and it sure seems like something kids would enjoy too. So for our upcoming shows in Juneau and Spokane, we have developed a visual scavenger hunt for gallery visitors to promote engagement with the work and also teach about the fertile biodiversity of the Alaskan deep. Participating is simple. When you visit the gallery show, make sure to look around for the clipboards and laminated lists of hidden organisms. Grab the wipe-off marker and take a gander around the gallery. Peer at the work. See how many shrimp you can see, or brittle stars, or even anemones. Write down your answers, take a quick photo of the sheet, and share it on Instagram or Blue Sky. It's sure to be a fun hunt, especially since there are loads of amazing deep-sea creatures to find hidden in paintings, illustrations, and even sculptures. So if you're free and in the Juneau area, we would love to see you at the opening on Friday, February 6th. Or if you can't make it in person, stay tuned to our socials, since we will definitely be there in person to share the fun. That's all for now.
As a little reminder, anyone who supports the show through the Patreon gets access to the Discord. Lots of fun, festive stuff going on over there. As we said, we had our Christmas holiday party where many episodes of Octonauts were watched and enjoyed, in quotation marks, by all, but we got to yell at them for a bit. The Deep Sea Pets channel continues to let us see animals which are definitely not deep sea, but we're bonding over our pets. All hail Cornflake, our cult leader, guinea pig. We talked about some of the animal saving videos as came up on Tyler's interview, the sewer end of YouTube, and the skepticism around some of those videos. Excellent dragon-based book recommendations. That seemed to be the theme of the recent books. Shared some Antarctic videos from Falcor. They put out a nice video from our cruise we did last year. And apparently people are planning to build a time machine to scuba dive in ancient seas and study the squid therein, and maybe go on a boat trip with Darwin. So as soon as that time machine is available, we'll make it open source. We'll do it like the data logger, and we'll just share the time machine. I'm sure that's not going to sort of tear reality apart if we each get a private time machine.
Yeah.
Photos of unimpressed squid interacting with the toilet brush of science, the TBS, which is actually a real thing held by the manipulator arm of ROVs, good for poking and annoying wildlife. Comparing holiday baking stories, photos and observations shared from some of the dive streams going on, and as always, various travel to aquariums around the world. Somebody goes on a jolly and they share it with the rest of the team, and we chat about some of their favorite animals. So we'd love to see you over there if you fancy joining the Discord through the Patreon.
Speaking of time traveling, I read something recently about, have you read this one about how 9/11 was a direct result of Kermit the Frog?
I've seen the Dominoes meme. Is it?
Apparently, I haven't seen the film, but apparently in the Muppets Christmas Carol, Kermit is shown, and this was made after 2001, I think. And Kermit was shown what the world would look like if he wasn't in it. And the world that he wasn't in still had the Twin Towers.
Because they used stock footage.
So something Kermit did led to the destruction of Twin Towers.
Oh, and on that bombshell, we'll deep see you next time, and we miss you already.
Bye.
Bye out.
Bye. Well, frogs are deep sea, so it's okay to talk about Kermit on the Deep Sea podcast, because we did figure out many, many months ago that there are frogs at more than 200 metres in Loch Ness.
I'm still not over that. That is wild.
Yeah. Frogs are deep sea.
The frog you can find under a rock in your back garden can go to over 200 metres. Good old common British pondy frog.
It was a Scottish frog.
That's the difference. Because I grew up in England, I see our soft English frogs, not a proper Scottish frog. Go to 200 metres at near freezing.
Yeah, and it's been there for a week, and it doesn't even need to be there either. It's
just doing it to look hard.
He's just doing it because his mate told him he couldn't.
That's it. That's the difference. It's because it's a Scottish frog.
Yeah, you're underestimating the Scottish gene.
Tougher.
Yeah,
not soft.
Tougher and don't necessarily think things through.
Stubborn might be said.
Yeah.